Laura is seeing patients and got involved, so she usually does the introduction. So but she can't today. So I'm going to introduce our speaker, Chris Posavad. Chris is the IDCRC laboratory in the laboratory operations unit. She's also the Senior Director for Faculty Affairs and Strategic Initiatives in the Vaccine Infectious Disease Division at the Hutchinson Cancer Research Center. She's also a Senior Principal Research Scientist at the University of Washington. Chris and I had an opportunity to talk a little bit about how she got to where she is, and hopefully she'll share a little bit about that. I think one of the things that I have always benefited the most from in clinical investigation is really having a close liaison with the laboratory, with making sure that that I understand the things that are going on, and also that I listen to what the laboratory scientists tell about what we could do in our studies. So I'm really, really pleased to introduce Chris. And without further ado, you're on.
Okay. Well, thank you very much for inviting me to present today. I'm very honored and flattered to be asked as the LOU representative, because as you'll see, my career has not been in lab operations, it's been in bench work. So it's been a real new experience for me to transition to this position. So let me see if I can just share my screen. I'm hoping this, let's see. Can you all see that?
Yes, it's very clear. Make sure everyone else who isn't speaking is muted.
Okay, so I thought I would just give some overview of some of the laboratory aspects of clinical protocol development. It sounds a little bit dry perhaps, but just please feel free to ask questions throughout, interrupt as needed. I want it to be, you know, as informative and conversational as it can be. The overview of my presentation, I really took advice from Kathy and Anna Wald and really thought it might be of interest to explain how I got here, and then to give an overview of the laboratory operations unit, some of the lab considerations for protocol development. I picked protocol development especially because that's what I've really been involved in over these last couple months. So I have some at least experience to speak of. I still don't have a lot to offer in terms of implementation and closeout, but hopefully at a future lecture I can provide some details about that. I also wanted to talk about our specimen sharing process, which I think would be of special interest to you all if you're interested in gaining experience in some lab work. So I was born and raised in Brantford, Ontario, Canada. So yes, I am Canadian. And I received my PhD in medical sciences from McMaster University at Hamilton, Ontario. And my thesis really involved the study of inhibition of T cell responses, especially CD8 T cell responses by herpes simplex virus. So here's my virus that I dedicated the large majority of my life to, as well as the T cells that are really the sort of the interface between the virology and the immunology is sort of where I see myself. After finishing my PhD, I thought it would be fun to go to the States to do a postdoc. And so I connected with Dr. Larry Corey in Seattle, and he was my mentor for my postdoc. And I received a fellowship from the Medical Research Council of Canada, which is now called the Canadian Institutes of Health Research. So basically Canada's NIH. And it was a 2-year fellowship, and I thought I'll stay there for 2 years, and then always thought I'd come back to Canada. I was a senior fellow at the University of Washington and a research associate at Fred Hutch. And my postdoc really focused on the characterization of HSV2 specific CD8 T cell responses in HIV infected people. I was also involved in some phase 1 immunogenicity testing of some candidate HSV2 vaccines, and I was able to secure a K22 to look at a unique population of people who were HSV seronegative but had T cell responses that we could detect to HSV2, sort of similar to the HIV exposed seronegatives. We described a similar group of people who were frequently exposed to partners infected with HSV2 but remain seronegative. And so, of course, Seattle is a great city to live and to raise children, and a real hub of infectious disease research. So it was very easy to make the decision to stay in Seattle. My first appointment was at the University of Washington in the Department of Laboratory Medicine. And my research really focused on CD4 and CD8 T cell responses to herpes simplex virus. We looked at responses in both immunocompetent and HIV infected people. We had an R01 for several years, at least a decade, to look at responses in co-infected people. I was interested in looking, at circulating T cell responses to herpes, but we also felt because herpes is it infects mucosal surfaces and reactivates in the skin, that is important to look at both circulating and local T cell responses. So we spent a lot of work using cervical cytobrushes and cervical biopsies to look at T cells in women who were infected, as well as, herpes reactivates at the skin. And so also biopsies of skin from reactive, from herpes lesions, as well as healed lesions. So we spent a lot of time finding new and more biologically relevant specimens to test to analyze the T cell response to HSV2. And then again, I also mentioned our unique population of HSV negative people who had durable T cell responses to herpes simplex virus. And I have now been involved in 2 or 3 at least of various herpes candidate vaccines. But as life goes on, sometimes you want to change, sometimes you want to use your experience in other ways. And so, as Kathy mentioned, I transitioned to a more administrative role as a senior director of VID faculty affairs and strategic initiatives at the Fred Hutch. And I am really oversee faculty recruitment, faculty review, faculty promotions within the division. And I've also worked with my colleagues to develop a mentorship program for junior faculty. And that's been really great. We have a really amazing group of faculty members in VID and at the Fred Hutch. And it's been a real honor to be able to help support them. I also work closely with Julie McElrath, who is the division director on VID strategic plan. So that's another area that I have worked on, which has really expanded my just my range of activities that I am able to be involved in. I'm also now currently just over a year, the Associate Director of the Lab Ops Unit for the IDCRC, and really thrilled to be working with the co-PIs, Julie McElrath from Fred Hutch and Mike Gale at the UW. And as I'll discuss further, we develop, implement, and evaluate lab research for infectious diseases as part of the IDCRC. And I also continue my appointment at the University of Washington, where I am involved with the HIV Vaccine Trials Network Laboratory Center, where Julie is also the PI. And I work closely with the team regarding mentorship and training in that capacity as well. So just to give you a brief overview of the LOU, we lead the development, implementation, and evaluation of laboratory research essential to the successful execution of the IDCRC research agenda. And our goal is to create an outstanding lab component for the IDCRC to complement the clinical and statistical activities. Here's our leadership group, the co-directors, Julie and Mike. And then for laboratory operations, I have the privilege of working with John Harrell, who is the director of lab ops for the HVTN, and Mike Steyerwalt, he is the QA manager for the HVTN and the LOU. And I have excellent administrative assistance from Megan Meager as well. And then Renee is the administrator for the LOU. And so for year one of the IDCRC, that was supposed to include time for development of processes and travel to the VTEUs and associated labs to really understand their lab capabilities. However, the pandemic prevented us from traveling, and the need to support urgent COVID protocols meant that we needed to develop processes as we were performing them. And as David Stevens would frequently remind us, we are building the plane as we are flying it, and truer words were never spoken, especially for the LOU. So please bear with me. We're still working out a lot of our processes as we go along. And I wanted to sort of just give you a sense of the types of responsibilities that are in our purview. We're really involved at all stages of the protocol, including development, implementation, and closeout, quality assurance. We have a deep involvement in specimen processing, handling, and shipment. We develop SOPs for all these processes and lab assays as required. We track the Duke IQA program, which labs, site affiliated labs need to be part of, and that really is to assess their proficiency in processing PBMC. We are involved in the selection of endpoint labs to support the per protocol endpoints. We work with the endpoint labs for budget requests. We're involved in immune monitoring, and we also have a lot of involvement in processes involving specimen management and working with the repository. And as many of you may know, for the clinical protocols, the sites need to utilize a lab information management system. And so there's additional complexity here because when MS is the data manager, they use Global Trace for specimen management, but SDSU uses the LDMS system. So it makes it more complicated than if both data centers use the same type of specimen management. So during my independent research program, specimen management really meant the clinic would collect samples, they'd courier the samples to the lab, we would process and freeze the samples, and then the samples would be thawed, assayed, the data analyzed, and published. Pretty straightforward process. But this has been a whole new world for me now in the IDCRC in terms of understanding the integration of the clinics, the specimen processing labs, the data management center, the repository, the endpoint labs, and the protocol teams. And so this is just a flow chart. I don't expect you to, it's a lot, but just to show the complexity of the specimen management process that we have been working on. This particular slide details our specimen management process when SDSU is the data center for the IDCRC clinical protocols. And there is a different process for MS. So again, just sort of outlining or highlighting the complexity for the specimen management for these clinical protocols. So the process involves the clinical sites up here, and they're collecting the primary specimens. They send those specimens to the site processing lab. They process, aliquot, and label, and they freeze, and they ship to the repository, which in this case is Thermo Fisher. So in a parallel way, parallel mode, the samples are then logged into LDMS, which again is the specimen management system that's used by the SDSU, and it's overseen by Frontier Science, and they they manage that part of the process. And so once all the samples are in the repository, SDSU then is involved in managing those samples so that when we're ready to select samples to be sent to an endpoint lab for their assay, the SDSU provides the pick list. Thermo Fisher pulls the specimens and they're shipped to the endpoint labs who receive the labs and again need to work to make sure that the specimens are received and logged in. And this, of course, this process here doesn't even, this just ends at the specimens arriving at the endpoint lab. There's also another process whereby the lab, of course, they perform their assay and then they need to work with the data management center in formatting their data and uploading it to the data center that will then analyze the data. And so we are still, between the LOU and the SDSU, refining this process, and it will be in effect for the first time in the MOMIVAC study, which is our maternal observational study, which I'll talk a bit more about in a minute, as well as the mix and match study that we're also working on in the IDCRC, and I'll describe that as well. So some of the lab considerations for protocol development in terms of the LOU, we consult on the overall trial design, including the study arms, the immunization schedule, blood volumes, time points, the types of specimens, making sure there's going to be enough blood at the right time points. We also review the objectives and the endpoints. Of course, most importantly, focused on the immunogenicity endpoints, both primary, secondary, and exploratory endpoints. We're involved in identifying labs and assays to support the per protocol endpoints. We work with the teams to draft the lab sections of the protocol and the MOP, most focused on specimen collection, processing, handling, and shipment. And we're also involved in drafting and designing the central assay plan, which I'll talk about in a couple of slides. Just wanted to explain a little bit about how assays are prioritized. Assay performance is critical to the successful product development and evaluation. And as many of you may be aware, there are 4 different assay development stages, including developed, optimized, qualified, and validated. They all require different, they have different requirements, which are laid out in the DMID guidance document that I'm happy to share if anyone is interested. For phase 1 protocols, which is where I've mostly been involved since I've been part of the IDCRC, the phase 1 protocols are powered for the primary endpoints. So there's a minimum requirement that assays supporting the primary endpoints are optimized, but we would prefer qualified and validated. And there are different considerations if the study is phase 1 IND versus an observational study. We also have to think about whether the labs that we pick are done if the assays are done centrally in a core lab versus a specialized lab. And we prefer central labs for the primary and secondary endpoints, where I think specialized labs are more apt, more appropriate for supporting exploratory endpoints or ancillary studies. And then the assays are selected, of course, based on the best scientific knowledge of the time. And as an example of this, I wanted to show a little bit of the work that we did to identify neutralizing assays for SARS-CoV-2 for vaccine evaluation. So we performed a performance and concordance study as part of the LOU. The background here, it's basically that the induction of anti-spike neutralizing antibodies is a major goal in the design of protective SARS-CoV-2 vaccines. and there was a need for a highly sensitive, specific, and high throughput assay that could measure neutralizing antibodies in response to vaccines. And this was to take place of the sort of gold standard, the plaque reduction neutralization test, the PRNT. So our objective was to bridge the PRNT assay to more high throughput assays and to inform further development and selection of assays for upcoming SARS-CoV-2 vaccine trials in particular. So we designed a study where we constructed convalescent serum sets, and we shipped these to various labs to evaluate the assay performance and concordance among the assays. And we picked assays. The FRNT and the PRNT are two wild type viruses, and those were conducted by Mehul Suthar at Emory. We used a reporter virus, the Nanoluke, in Ralph Barrick's lab, and Mehul also has developed a FRNT with a neon green reporter. And then we also worked with David Montefiore at Duke using his pseudovirus using full-length spike. And so this is like more data than you want to see, but just to show you that the SDSU, we worked closely with them, and they performed a concordance analysis across all assays. And you can see here, this is the concordance correlation coefficient. And so you can see these assays are most highly concordant, and they're listed by concordance here. So you can see how we compared the data between all the different assays here to look at how closely they were related. And I can summarize these data on the next slide. And what we showed along with the SDSU was that the FRNT that was run by Mehul Suthar at Emory showed the best performance among the live virus assays. The pseudovirus and the FRNT also showed equally high levels of performance, and both were superior compared to the PRNT. The live and pseudovirus assays can be highly concordant, and performance can significantly impact concordance. We also showed that whether the spike variant using 614D or G, that can definitely impact concordance as well. And the final result was that the FRNT MNG from the Suthar lab was selected to bridge the PRNT in the Moderna phase 1 trial due to the excellent performance and the high throughput. And so, is there a question? Okay.
Sorry, I've got a question. Might just reveal my ignorance, but would that be okay?
Sure.
Would concordance of 1.0 be a perfect concordance?
Correct.
Okay. Can you just show us two slides back again? What? Okay. And which one of those was so the top one was selected as a good concordance? How is a good concordance defined? What's the threshold?
Yeah, that's very that is beyond my this is all work by the SDSU, and I think, like you were mentioning, like, you know, one is the highly concordant would be at the 0.99 range. We were hoping that we would receive, we were hoping the assay, so what am I trying to say? There was no real baseline or benchmark for this because these assays were new. And I think the idea was the performance of both of these assays, the FRNT assays in terms of linearity and sensitivity and specificity, were all taken into account for the concordance concordance correlation coefficient, the CCC. So it takes into account numerous different parameters regarding the performance of the assay. And so both of these assays, the FRNT and FRNT MNG, were both highly correlated. And that's why, because the FRNT MNG was actually higher throughput than the FRNT, was the reason that we moved forward with that assay to support the Moderna studies.
Okay, thank you.
You're welcome. So like I said, we selected Mehul's FRNT MNG assay due to performance and concordance, and due to the fact that it was high throughput, which is needed in these phase 1 studies where people want the data yesterday. And so based on that, he was selected for the live neutralization assays for this trial. These were conducted in the older cohorts between 56 and 70 years of age. And his work has subsequently, or his assay has been subsequently selected for additional upcoming trials, including the mix and match and the MummyVax trial. So Mehul's done a lot of work since then to continue to develop his assay, where it's now more at the level of qualified. So yeah, so that's just sort of one of the real world examples of sort of what we have done and how we've selected assays and assessed their performance for these trials. Another thing that we have worked on, which I think is new, at least to the MS process for specimen shipment and specimen triggering during a clinical trial, is to develop a central assay plan. And everybody, of course, is very anxious for clinical trial data. So it's really unacceptable to wait until the study completion for all the data to be analyzed. So it's preferred that there are interim data reports generated on either full or partial enrollment and on select key time points. The time points can be selected based on peak immune responses, or time points can be selected to assess the durability of the response, for example. So we have adapted a plan that the HVTN uses and now have worked on it as a template for the IDCRC. And the Central Assay Plan, or the CAP, summarizes the plans for testing specimens to assess immunological endpoints in support of per protocol objectives. It describes the reports or the data sets that will be distributed, including the specimens to be shipped and contributed, and that will contribute to each of the report. And each assay, if you look at a B cell assay or a T cell assay or an antibody assay, they each have their own specific plan laid out in the Central Assay Plan. And it really, some of the things that go into the specific plan includes the preferences of the endpoint lab for specimen shipment. Some want the specimens shipped, weekly. Some want them, batched in a different cadence. So we work with each of the endpoint labs to make sure that their requirements are met. There's, of course, different immunological time points depending on what you're assessing. If you're looking at an antibody response versus a T cell response versus an innate response, those would all be unique to the assay, as well as the number of time points and the types of specimens that will be evaluated. And so I wanted to give just a sort of example on a central assay plan that we are currently working on for MummyVax, which is a 21-004 IDCRC DMID trial. And MummyVax is the multi-site observational maternal infant SARS-CoV-2 vaccine study. And as the title notes, it's an observational non-interventional prospective cohort study to evaluate immune responses in SARS-CoV-2 vaccinated pregnant and postpartum individuals and their babies. Group 1 are individuals who receive SARS-CoV-2 vaccine, the EUA dosing during pregnancy. Group 2 are individuals who are vaccinated postpartum. And then group 3 and 4 are the respective babies for cohorts 1 and cohort 2, or group 1 and group 2. And so here's a table that outlines all the samples for the primary and secondary endpoints for this trial. So here are the cohorts. We look at the women or their babies. And as I mentioned, there are 2 groups for the women, whether they're vaccinated during pregnancy or postpartum, and then their respective babies. We are looking at testing women who receive up to 5 different vaccine platforms. We're looking at Moderna, Pfizer, and J&J right now. And if additional vaccines receive EUA, we will include those in the study as well. We're looking at enrollment of 150 women per vaccine platform and their babies for women who are vaccinated during pregnancy, and 50 per vaccine platform if they receive the vaccine postpartum. And you can see here for the primary endpoints, we're looking at both serum, and that would be from venous blood versus cord blood. And we're looking at binding antibodies and live and pseudovirus neutralizing antibodies in these 2 specimens. And then a secondary endpoint is the evaluation of breast milk, and we'll be looking at IgG, IgA, binding antibodies, as well as live and pseudovirus antibodies in breast milk. And so here just lays out the different time points on the total number of visits for each of these groups, the total number of samples, and of course with cord blood, it's just at birth. And so we start with this table looking at all the potential specimens that will be collected. And from there, we make assay-specific plans on how we are going to release the data as it's analyzed. So here is just an example of the cap for serum binding antibodies and from antibodies from serum. And it's using the MSD platform will be employed by Marcella Passetti at the University of Maryland to support this primary endpoint. And she'll be looking at spike and RBD serum IgG antibodies. And so here lists the number of interim data reports that we're expecting and what the report content is. So you can see here, the first report will be the first key time point in each of these different vaccine platforms and the number of samples that will be shipped to the lab. And after all the participants have reached the time point at day of birth, then that will trigger the specimens to be shipped to the endpoint lab. That will then run the assays and then upload that data for analysis, and that data will be shared with the protocol team. So as I mentioned, they don't want to wait until the end. So our last time point is 12 months, and no one is interested in waiting that long for data. So this is sort of just an example of the type of, process that we're using in order to trigger shipments so that we can have these data to share with the protocol team. And then just as a, to give you just a difference, now we're looking at breast milk, same binding antibodies, but in a different specimen. Of course, there are different time points selected because it's looking at, transfer of antibodies to breast milk after moms have been vaccinated, and they will have a different plan. So if there are any questions, if not, I'll move on to the last part of my talk, which is really just to describe the LOU's commitment to mentorship and training the next generation of researchers. And in fact, our specific aim number 4 is to mentor and train the next generation of researchers. Our plan is to foster new leaders in clinical infectious disease research. We'd like to offer opportunities in training and lab assay development and application. We're interested in developing a scientific exchange program between the LOU labs and external labs so that trainees and early stage investigators have an opportunity to work in labs, develop collaborations, and develop experience in performing clinical research. We also will be providing access to unique specimen sets from IDCRC research projects, and this will also help to increase the laboratory capacity in the IDCRC and the VTUs, which is again one of our goals in the LOU. And so one way I think that we can best help with or support this aim, achieve this aim, is as part of the ancillary study request process. So we're currently drafting the process for sample sharing, and this will require an approval process as outlined by the leadership group for the IDCRC. But currently, our draft proposal, we'll create a protocol-specific ancillary studies committee, and that'll include the protocol chairs, an LOU rep, an SDSU rep, a regulatory rep, and then DMID and an industry rep if applicable. And the committee will receive proposals from investigators, and we're working on the process for investigators to submit these proposals. And the committee will determine, are the specimens available? Is there overlap with the proposal for the per protocol endpoints? And will the study add value and be scientifically impactful? And if all these are a yes, then we'll work to make sure that the proper regulatory activities are approved. We have to make sure that the participants have consented to future research studies and make sure any material transfer agreements are set in place. And then we can release those specimens to the investigators who will then provide a written report and are able to publish and present the work. So I think that would be a really great opportunity for anybody who is interested in these very valuable specimen sets, as well as wanting to have an opportunity to gain experience in laboratory assays. Some of the current protocols that we're working on are the Gritstone Oncology company that has developed SARS-CoV-2 vaccine using a adenovector and mRNA, so heterologous vaccine platform. They also are, it's very interesting because they have included a T-cell epitope cassette as part of their vaccine strategy. And so we'll be collecting serum, plasma, and PBMC, which can be available in an ancillary study request. We also are working on the Moderna trials, so the original Moderna 2003, as well as the boost sub-study. So the original Moderna vaccinees can receive a third 1273 boost. We're involved with the Moderna variant, so really boosting with the South Africa B1351 vaccine. And then our mix and match is the heterologous vaccine platform using Moderna and Pfizer and J&J. We haven't started enrolling in that, but we will soon. And again, that will be a lot of specimens available. And then, as I mentioned, sort of the one most near and dear to my heart is MummyVax. And we will be collecting maternal and infant serum, cord blood serum, and breast milk. So really just a treasure trove of specimens. And I highly recommend development of proposals that can add value to the protocols and provide you with valuable lab experience and make new collaborations as well. So just a couple of takeaways. Maybe this is obvious to some, but I'm learning R01 research is not like network research. There are a lot more restrictions and requirements. There are a lot of meetings, and as someone from the West Coast, it's been very important that I become a morning person. I have a lot of early meetings, so that's been a wake-up call for me. I have gained a lot of experience now in team science, and it's been a really incredible experience for me. It's been a lot of work, especially with the COVID protocols, which are all fast-tracked, but I have just had, I've had an amazing time. I've met amazing people, and it's just been a real privilege for me to be part of these protocol teams. I've also noted that the networks also have different processes and procedures. So what I've learned from the HVTN isn't necessarily the process or procedures that will be implemented for the IDCRC. So just a lot of complexity and a lot to learn. The mentor can be the mentee. So even though I'm working with our early stage investigators, through the Fred Hutch on our mentorship program, I am being mentored myself, especially with my lab ops experts who have just been invaluable for me in terms of helping me get up to speed and really helping the LOU get on its feet. And I just would really encourage you to consider proposing an ancillary study to access some of these very rich specimen sets, and mostly because lab work is really fun. So I would just like to thank a lot of people. First, my team, the LOU group, and especially John Harrell and Mike Steyerwald, who have been my mentors during this process as we're getting the LOU up and running. We've collaborated closely with the SDSU and Elizabeth Brown and her team. Of course, the support from David and Kathy in the leadership group. Just excellent collaborations and support from DMID, especially Chris Roberts, Catherine Luke, Christina, Marina. I mean, just been really, really great. The leadership group, the COU, of course, FHI 360 has, and they've been, incredible as we work through all of our processes and how we integrate our different processes. The VTUs, too many to name here. I'd like to thank Kathy and Anna just for their advice going into this lecture. And then just to note who the labs who helped us during the neutralizing antibody studies, as well as the protocol team for MummyVax. And
that was terrific. I'm going to need to run a little bit early. So Iko, could you sort of orchestrate the questions? That was really wonderful. I did leave a question there and I have to go pick something up I forgot to pick up. So wonderful. You guys stay on. Iko, you're good?
Yeah, I'm good.
Okay, thank you.
Thank you, Chris. This is just really amazing, especially for highlighting the opportunity for our junior investigators to be engaged with the lab. So we're going to open this up for questions and discussion. Does anybody have a question? Let's see. So there's a question on the chat box from Kathy. How can mentee propose ancillary studies? Do they need to work with their PIs, or are you available to talk about this with the individual mentees?
Yes, I'm absolutely available to talk about it. We're in the process. We've developed a process document, and I literally yesterday just sent it to the leadership group. And then there's a process where they'll need to weigh in on what we have proposed. And currently there really are no specimens for the LOU under our purview yet, because we're just in the process of getting these protocols up and running. So the table that I had presented regarding the Moderna trials and that those specimens will be available, and I would be happy to help. And we will be sharing the process more widely through once it's approved, we'll be sharing this process to make everybody aware of what's required. But we're trying not to make it too onerous on people, but we're, it's again, something we're very, it's really important to us that people, especially trainees and early stage investigators, have the opportunity to access these samples. So we could help, in drafting the proposals and, in explaining the process for approval.
Yeah. So I just have another follow-up question. So access to specimen from studies that are sponsored by industry, like Moderna, for example, is that something that our investigators will have access to? And then the second follow-up question is whether your lab can support, say, junior investigator, for example, who needs to run an assay that is specialized for their study, can they support those assay on a fee for service basis?
That's a great question. I don't know the answer to the second question.
Okay.
But for the first question, I mean, all the specimens are available. They just need to, there has to be an approval process. So I have not been part of the process for obtaining specimens from the Moderna trials, for example, but there is a specific committee that, and I can certainly provide the connections there on who is involved in moving those requests for specimens through the process and hopefully being successful. I think it's, when I think wanted, some advice that I would give is if you are at all interested in the Modernas that are currently happening or any of the vaccine trials, to get your request in sooner than later. You don't have to wait until the trial, the specimens are there. There will be plenty of specimens left over. And so one thing that came up was, well, how do people know what specimens, like what time points? And so I, again, haven't figured out sort of all the details in terms of whether we can share these protocols so that the investigators can see the various time points and specimens that are available that they would want to request and propose, a study. But we're definitely working on it, and the plan will be to share it more widely, and we'll probably present it at some of the meetings like the VTU, PI meeting, things like that, so that there's general awareness of the availability of these samples.
Awesome.
Hi, this is Erin. Thank you for the excellent talk, Chris. It was wonderful. And I have like 3 little questions, so I'll take one at a time, I guess. So one, if we have questions about whether our assay is developed, optimized, qualified, or validated, because that document is a bit, I think there's some ambiguity about it in certain areas. And so is there someone we can reach out to with questions about that?
Yeah, I definitely, we definitely have experts here. I am not one of them, but John and Mike are definitely, and that is part of our list of responsibilities to really help labs and understand the development and what they need to do to have their assays become developed, qualified, optimized, and validated. So you can contact me, Erin, and I can make sure to connect you. But I agree with you on the guidance document. I'm not sure if that's something that we're, in the long run, the LOU will want to work with DMID to update so that it's a bit more clear, precisely, what's needed. So I definitely think that's a gap that we have not yet addressed because we're just trying to, keep our heads above water. But I definitely think that's a really important function that we could, do to help the labs really understand, assay development so that they can be included to support primary and secondary endpoints.
Okay, thanks. And then a question. So I recently went through a process of requesting samples for Act 1, and it went through a DMID review process, or actually it may have been NIAID review process, and I was given approval to have the samples. They're sending to us with MTA.
Oh, great.
So that's great, yeah. And so I just wanted to ask about this because, like, for them, the MTA process is very straightforward, pretty easy, and like all they want is an acknowledgement for the samples. And then it seems like for this case, I don't know yet about the MTA, but it seems like with reports and publications and presentations and stuff. So my question is, in general, like, how are you guys trying to balance like the access to these samples for good science versus like, potentially like the paperwork load that might present a barrier, or like if there's restrictive MTAs? Do you understand what I mean?
Yeah, we definitely don't want it to be a barrier. I think one thing that we're interested in is just making sure there's some accountability to the specimens. So the idea of having a written report, whether there'd be an interim report or we really just don't want to have these samples go and then have no idea, what happened to them. I do think there should be more streamlining of the MTAs and transfer of samples, because I know we've worked on different, you know, umbrella MTAs. But again, I've only worked with this like a couple times, and it didn't seem to be that onerous for us to obtain samples. By the way, we couldn't get Act 1. We got Act 2. So go, go you. We couldn't get those Act 1 samples. So I think we don't want it to be onerous, and we don't want it to be burdensome, and we want to be able to provide those samples. But I don't have the final details, because again, I'm not in the weeds on regulatory. And so I think that's something that I'm learning is missing, and just trying to figure out how we can fill the gap so we have a regulatory expert who can really help us navigate those types of concerns.
Great. Yeah, because I've had throughout the COVID process, I've had NTAs with all sorts of people, and one of them was with IAVI. And for example, like we have to provide, you know, drafts of manuscripts like very far in advance, and then they have to be approved and all this stuff if we want to. So there can be those MTAs, and there could be MTAs, which is like, as long as it's at Emory, we're fine. If you share it outside of Emory, it's a problem, right? So that's like a more like kind of thing. Anyway, so it's, yeah. And the last question is on mommy vax, just a very tiny baby question, but are you guys looking at IgG subclasses? Because that's important for effector function and clearly what's going from the moms to the babies.
Yes,
is that being
yes, they definitely want to do that. It's not a primary endpoint, but it is, we are, and we will, and someone will do it, and we haven't gotten that far yet. I was showing you what we don't know yet. We definitely know that we need to look at that. We also talked about the FC glycosyl, you know, your studies as well. So I feel like there's this, you know, list of really exciting studies that can be done, but our focus right now is just on the primary and secondary endpoints. But yes, and we will keep you informed on that one.
Good. More questions, comments?
Yeah, I have one more question. Thank you for the presentation. That was really, really nice. I have actually a couple of questions. So if we decide to request samples, do you have a process in place that involves, I don't know, like forms, fill up forms besides the proposal, or any particular structure proposal in a definite format, or what is like the details or requesting samples? And maybe to that same question, when we request samples, do we get access also to some clinical information, or it will be just the time points that you were talking about? Thanks.
Right. So again, we don't want it to be an onerous process at all. We want to understand what the, you know, the goals, what is the, you know, the detail, the research plan, so that we can assess that it doesn't overlap with what is going on in another study that's been approved. We would need to know the time points, what types of specimens, how many specimens, like how many participants you would need. If you've worked with a statistician to discuss sort of power calculations, things like that, so that you know, so that the idea is these specimens are very valuable, and we want to provide, but we also want to make sure that the data will be interpretable and robust. Let's see, what else will we need? I blanked for a second. What else did you ask me?
I guess if there is any special format that we have to put the submission on, or it will be the usual IDCRC kind of format.
We don't have that template yet, but there will be a template. And it will basically say, you know, blurb on what the research strategy is, what specifics of the samples, what's the study, how many time points, how many vials per time point, what type of specimen. And then the funding, we have no guidance yet. And I think that's something that's also been worked on, or that we're working on. But we definitely want to make sure the template is clear and not burdensome. And and then we're still, like I was mentioning with Aaron, just trying to work out the regulatory aspect of it and needing that expertise on the committee to really understand, you know, make sure that we're, you know, we're following all the proper guidelines. But I'm really glad to hear that you're interested in accessing these samples. And I think the sooner that you think about proposals, like I said, I don't think you have to wait until all the studies over to be able to propose these. And I'm really excited to get you the details of this process so you all can start thinking about the types of studies you'd like to propose. But we're here, we're advocates for you and wanting to, you know, to get you these specimens and help whatever way we can. If that's connecting you to a lab that is doing something that you're interested in bringing back to your own lab or whatever. So I think all of those things are on the table, and we'd like to do what we can to help facilitate those interactions and collaborations.
That's great. And I'm sorry, it's to follow with that. Is there any restriction in being a participant in the study, not me myself, but like our site, to have access to those samples? So for example, I'm with NYU and we will be doing the mix and match, but we did not do the Moderna trial. So do we have any restriction of the samples that we can potentially access? I guess if there, I'm not sure if there is any conflict if we request access to samples of the studies that we are doing in this site, or if it will be better to just have access to samples that we don't do, we're not doing the studies in our sites.
I don't think you have to be contributing to the protocol to be able to access samples. So that's not my understanding. Now, I don't know, again, what the restrictions are. If it's a Moderna trial, like a company sponsored, I'm still trying to understand like who owns those specimens, because on some calls, it seems like the site that collected those specimens own the specimens, but then in other studies, the industry owns the specimens. So I haven't quite figured out that yet. But in terms of the IDCRC and our the protocols that we're supporting, you do not have to be participating in that protocol as a site to be able to access those samples. Now the proposal has to be approved, but I don't think there's any restriction there. And I think you asked me also about clinical data, and I think part of the template will include what data do you need. So you would be able to, you know, request specific clinical data if that's what is required for you to do your analyses.
Very good. Any last burning question before we wrap up this session from anyone? Chris, you did such an amazing job. We're going to ask you to come back again next year.
Well, I'll know more about protocol implementation, so to be determined once I actually go through the process. So.
Yeah, you made a very complicated process look so simple and straightforward.
Oh,
Thank you so much. We really appreciate it. And so we'll keep in touch, and I'm sure you're going to be hearing from several people on this call and those who are not on this call. Thanks.
It's really been an honor to present for you, and I really look forward to working with you and hoping to see your very exciting proposals for specimens. So
wonderful. Great. All right, you all have a wonderful day, and we'll see you again in a couple of weeks or so.
Great.
Bye, everybody.
Bye.
Yeah.